Hip, hip, hooray for DNA. It provides the key to the plans for making everything in you and me.
Welcome to one of the past HSC exam question videos. What we're going to do in this video is we're going to cover this exam question, which comes from the Biotechnology chapter. While doing a second, I'll read the actual question. Once I read the question, you get about 5 seconds to pause the video. Once you pause the video, attempt the question, and then press play when you're ready. The question itself has 2 parts. The first is cloning techniques that can be used to increase numbers in endangered species. What effect will cloning have on the genetic diversity of the species? And that's worth 2 marks. And B, explain 2 possible evolutionary effects of a disease entering an endangered population containing some cloned individuals. And that's worth 4 marks. So when you're ready, press pause and attempt the question. Alright, welcome back. So for the first one, it's worth 2 marks. So what you should write is you should quickly talk about what cloning actually means. And cloning means we have all of them being identical. And what you can then talk about is 2 scenarios if you have the time. I think I only gave 1 scenario in my answer, but you should have had 2 scenarios. One would be what happens if maybe the population itself was made up of mostly cloned species. So if that population was mostly cloned species, or if there were some clones and some non-clones. So some clones plus some non-clones. What would happen to diversity? So what I wrote here first is cloning techniques are a form of asexual reproduction, which means that the parent and offspring will be genetically identical. Right? And then I wrote, if a population consists mostly of cloned species, then the genetic diversity is minimal, it's non-existent, as all species within that population have the same genes and DNA. Right? So there's going to be no genetic diversity if they're all clones. And what you can write then as well, if you wanted to add some more, you can talk about what happens if it's not mostly, but there's only a couple of clones, but quite a few non-cloned. What would happen to genetic diversity? And in that case, it would also decrease. Generally, genetic diversity decreases because cloned species just mean that they are all identical. So the gene pool will have less different types of genes, no matter what the example is. And for B, explain 2 possible evolutionary effects of a disease entering an endangered population containing some cloned individuals. What you should write for this part is you obviously need to explain. So explain 2 possible evolutionary effects of a disease entering an endangered population containing some clones. So in this case, you would first say, well, if the cloned species are immune to the disease, what would happen? And if the cloned species were the ones which were non-immune or not immune to disease, what would happen? What would be the result of the evolutionary effects of this? So it might sound a bit confusing. I'll go over the actual answer and it should be more clear then. So cloned animals have identical DNA. So if one cloned animal is resistant to disease, then all others are too. Right? So if one of the cloned animals is resistant, that means all of them are. And the reason why is because they have the same DNA. So if cloned animals are resistant to disease, but the others are not, then the genes of the cloned animals will eventually dominate the gene pool as the other individuals have died. So here I talked about the first scenario. If the cloned species are immune, but the other members of that species are not immune, in that case, cloned animals will all survive, whereas the other members of the population will all die. And what that means is that eventually only the genes of the cloned species will dominate the gene pool. And that means there's going to be very limited genes because obviously they're all going to be identical. So that would be path 1. So we said the 2 possible pathways. Pathway 1 is if the cloned species are immune and the other ones are not, then you would have the gene pool being dominated by the cloned species DNA, cloned animals DNA and genes. And the other example is if the cloned animals are not resistant, but some of the other members of the population are, then the cloned animals eventually die out because they are not resistant, and their genes would no longer be part of the gene pool. The evolution of that species would thereby not be affected by the cloned members of the species. And so in this case, you would have the cloned ones just dying out, and everything's, in terms of the gene pool, everything's as it would usually be. But in example 1, we have a gene pool which is only from the one identical cloned animals gene pool, because in this case the cloned animals have survived, whereas the second example, we have a gene pool which has a lot of variation because the clones have died out and the normal members are still around, so the gene pool would be having quite a bit of variety. And that you would get 2 marks for the first and then 2 marks for the second, just briefly describing the different types of scenarios. And that would be a 4 mark out of 4 question. Whether these questions come from, they came from these dot points. Identify how the following current reproductive techniques may alter the genetic composition of a population: artificial insemination, artificial pollination, and cloning. And discuss the potential impact of the use of reproductive technologies on the genetic diversity of species, using a named example of a plant and animal that have been genetically altered. Right, so the questions came from those 2 dot points, but I hope that was useful.
Thank you for watching.